Okay, so now it is my pleasure to introduce Dr. Millie Dugal, who is a health science policy analyst from our Office of Medical Policy, OMP, within CDER. And Dr. Dugal will be our moderator and host for the next session. Thank you.
Thank you, Brenda, for that lovely introduction. Hello, everyone. I'm Millie Dugal, and I'm excited to be here with you all today. We have two lectures lined up for this afternoon's session. The first is on new trends in trial designs. Leonard Sachs will discuss decentralized trials and the role of digital health technologies in clinical care. The second presentation for this afternoon will be on informed consent. And Meeker-O'Connell will discuss informed consent and ethical considerations in clinical trials with us. So, without further ado, I would like to hand it over to Leonard, and I will be back with you all for the question and answer panel. Thank you.
My name is Leonard Sachs, and I'm going to spend the next half hour discussing innovative approaches to clinical trials. Why bother? Well, the boy in the wheelchair has Duchenne's muscular dystrophy, which, as I'm sure you know, is a devastating X-linked condition affecting the normal production of dystrophin. The condition causes progressive muscular weakness and eventually respiratory failure, with patients usually dying in their late teens or 20s. He's participating in a clinical trial of a new drug, and the trial site is at a pediatric neurology unit in Boston. To get to his trial visits, he has to leave school. His mother has to take time off work and arrange childcare at home. They take a 4-hour flight together to Boston, struggle to get into the Uber in the wheelchair, make their way to the hotel, and the next day they travel to the research institute. They wait for an hour in the waiting room and finally get to the research nurse. She supervises a 6-minute walk test, draws some labs, and does a quick physical exam, and they begin their long trek home. There must be a better way to do this, and that is the focus of my lecture. Technology-enabled clinical trials offer lots of new opportunities. As we all know, there's been enormous progress in communication technologies and telemedicine, which is widely used, in the ability to share healthcare data, images and documents, and the development of biosensors to record physiological features from patients. There are also opportunities for remote data acquisition in clinical trials, which are remarkable. They include getting data from participants, getting data from healthcare providers, and even from healthcare institutions. In September this year, we published our final guidance on conducting clinical trials with decentralized elements. Decentralized elements allow trial-related activities to occur at locations other than traditional clinical trial sites that are convenient for trial participants. This can be, for example, in patients' homes or local clinics and facilities. Why are regulators interested in decentralized trials? Well, first of all, accessibility. These trials provide accessibility for patients with rare diseases who couldn't get to trial sites. They provide accessibility to patients with mobility or cognitive challenges, and they provide accessibility to a diversity of participants from different socioeconomic or cultural backgrounds who formerly did not participate much in clinical trials. They improve convenience for patients. They provide other efficiencies. For example, they save on travel, on the need for physical facilities when activities take place at patients' homes or local clinics. And they also take advantage of community healthcare providers. And of course, we can avoid the transmission of contagious diseases such as we saw with COVID. Decentralized trial procedures are not new. In outpatient trials, once participants leave the study site, many trial-related activities take place remotely. For example, there is self-administration of investigational products. Adverse events occur in patients while they're at home and they present to local clinics or emergency rooms. Patient diaries have been used for years to document certain symptoms, such as seizures, such as pain, etc. Interactive voice response systems are traditionally used in many clinical trials. Telephone follow-up is often a feature of long-term follow-up in a trial, and electronic informed consent is fairly widely used since we published a guidance on this in 2016. Decentralized clinical trials involve a bunch of different strategies to take trial activities to the patient. In the middle, you can see a participant. The woman has paroxysmal atrial tachycardia, and you can see on the left she has remote telehealth visits with the investigator. She signed her electronic informed consent electronically. Her drug is delivered to her home. She goes to the local clinic for her blood work. She goes to the local healthcare provider for a clinical examination. And you can see the last example is a digital health technology that she wears. It's a mobile cardiac monitor that reports episodes of atrial tachycardia. Let's talk a little bit about remote trial visits. These are trial visits where the investigator can supervise remotely. And these are commonplace in telehealth, but they have certain challenges. For example, they have to be in line with local regulations on telemedicine. You can't do a physical examination over the phone. Video photography may not fully capture the features of a lesion if that's what you're using. Patient engagement in the absence of in-person contact may be a challenge. Complex drug administration procedures are difficult to do over the phone or over a video call. And in situations where you need close medical supervision, e.g. for infusion reactions, having the patient do the visit remotely is probably not suitable. What about the use of local healthcare providers and facilities? There are resources and qualified healthcare providers in the clinical care environment who may be able to be used in clinical trials. And there can be delegation of routine clinical activities to patients' local clinics or to healthcare providers for routine clinical procedures, for example, x-rays, clinical examinations, lab tests. It's important to ensure that providers have the appropriate qualifications, and it's important for investigators to review the data regularly to make sure that it's complete and correct. Home visits are a novel approach. They may either rely on dedicated trial staff or contracted healthcare providers going to patients' homes to do certain trial-related activities. Mobile trial units in vans are currently being developed by many sponsors, and this is another way of getting trial activities to the patient, although obviously the perimeter is limited. And all these approaches are designed to extend the physical reach of the trial. Investigational products can be directly distributed to patients. It's important for the investigator to control the release of the product to the trial participants. Local state laws differ on direct distribution to patients. They may require locally licensed healthcare professionals or pharmacists, and this should be clarified before the trial is underway. Packing and handling are important considerations to make sure that the product arrives in good order, and there have to be plans for patients to dispose of unused product. Electronic informed consent, as I mentioned, has been referred to in a previous guidance published in 2016. It's already in widespread use. It allows patients to review and sign informed consent at home. It may provide videos and graphics to make the process more informative and more easily understood. But of course, remember that signed consent must be obtained before enrollment. When considering whether an investigational product is suitable for decentralized clinical trials, the nature of the drug should be considered to determine whether administration outside of a clinical trial site is, as part of a DCT is appropriate. One should take into account the complexity of administration, the safety profile of the drug, and the stage of drug development. Drugs in early development are probably less well understood and probably not suitable for administration in a decentralized way. It may be appropriate for local healthcare providers or trial personnel working remotely to administer the investigation product at local healthcare facilities or at participants' homes. Remember that hybrid DCTs using both local and central visits may be considered for drugs that require supervised but infrequent administration. Investigator oversight. Investigators have several responsibilities. Firstly, oversight of the trials. They have to ensure that data provided by local healthcare providers is reviewed on a regular basis. Investigators should make sure that the data they receive are complete and consistent with the requirements of the protocol. And it's the investigators who will need to make decisions about whether a patient's complaint to a local healthcare provider represents an adverse event, whether a finding reported by a local healthcare provider represents a study endpoint, or if a protocol-directed change in treatment is necessary, because it's the investigators who are familiar with the clinical protocol. As far as patient retention goes, when patients are seen remotely, the jury is still out. We don't know whether patients are less likely to follow up, and this is something that we're anxious to find out. Investigators are also responsible for ensuring data privacy. A couple of thoughts about safety assessments in a decentralized clinical trial. Trial participants must be able to contact trial personnel to report adverse events and to have pertinent questions answered. Trial participants should be able to arrange for unscheduled visits using telehealth or an in-person visit as appropriate. If significant safety risks emerge because of remote administration or use of an investigational product, sponsors must discontinue the remote administration or use. And if authorized in the protocol, routine safety monitoring involving lab testing and imaging may be performed using local clinical lab facilities. Protocols should specify how adverse events will be identified remotely and will be evaluated and managed, and how care will be provided for adverse events that require urgent or in-person attention. Sponsors also have responsibilities in decentralized clinical trials. These responsibilities are the same for DCTs and for traditional site-based trials. Sponsors should strive for diversity and inclusiveness in trial populations. Sponsors need to account for multiple sources of data collection in a DCT and the data management plan, and they should describe in the trial protocol how the operational aspects of the DCT will be implemented. Some important considerations for decentralized trials are firstly that not everything can be accomplished remotely. Many decentralized trials will end up as hybrids, combinations of remote visits and visits to investigator sites when procedures such as detailed physical exams and other in-person activities are required by the protocol. To turn to a different sort of trial, this is a guidance that we've just published called Integrating Randomized Controlled Trials for Drug and Biologic Products into Routine Clinical Practice. These are trials which are sometimes known as point-of-care trials or pragmatic trials, and the idea is to conduct the trial in a clinical practice setting where the patient normally gets their medical care. The medical literature abounds with these sorts of trials, point-of-care trials, pragmatic and large simple trials, and what all these trials have in common is they rely on integration of clinical research with clinical care. Now, these approaches have not been significantly adopted for regulatory submissions. A poster child for one of the trials conducted in clinical practice was the recovery trial, which was done in the UK for COVID-19. This trial reportedly recruited 40,000 COVID patients through the National Health System in the UK within 6 weeks, and they were able to show the mortality benefit of steroids, tocilizumab and baricitinib in treating patients who were hospitalized with COVID. So practice settings allow for engagement of large numbers of patients in short periods of time, and these trials reflect the effectiveness of treatment in real-world environments. Accessibility of clinical trials to patients who wouldn't normally participate is another feature of these trials. To integrate randomized controlled trials into clinical practice and to conduct them where patients get their routine care, it's important that trial designs and activities are streamlined to align with clinical practice. These trials leverage established healthcare institutions and existing clinical expertise in the medical community to reduce startup times, speed up enrollment, and improve accessibility and convenience for patients. Real-world data from electronic or other healthcare records may be used in these trials. Trial-related activities may be conducted as part of routine practice with participation of local healthcare providers in these healthcare institutions, and dedicated trial staff may participate if needed to perform activities that require research-specific expertise. Mentioned earlier on, digital health technologies may also play an important role in decentralized clinical trials. In the context of clinical trials, we are interested in digital health technologies such as wearables, interactive applications, and instruments placed in the patient's immediate environment that measure a clinical feature of interest in a clinical trial. These may be instruments like smartwatches, cell phones, mobile glucose monitors, etc. This again is a child with Duchenne's muscular dystrophy, and he's doing the 6-minute walk test to evaluate drug efficacy. This is a clumsy test. He has to walk as far as he can within the space of 6 minutes. What if he has a sore foot or had a bad night's sleep, or if the supervisor of the test motivates some kids more than others? There must be better ways to measure this. This picture is of an accelerometer. And accelerometers are versatile tools for Duchenne's and many other diseases. Accelerometers are used all over. They're present in our cell phones, smartwatches, and Fitbits. Triaxial accelerometers give good three-dimensional pictures of movement. They can be very helpful in measuring steps or other activities taken by a Duchenne's patient. There's no need to depend on snapshot measurements and clumsy tests like the 6-minute walk test. Measurements can be recorded over long periods of time, so we don't rely on single measurements, and comparisons with baseline performance can be made. Accelerometers are not the only sort of digital health technology. Here's an example of several. For example, galvanometers, which measure electrical current, may be used. The output of these is typically voltage, current, or impedance, and they may be used, for example, to measure heart rhythm. There's an interpretive algorithm, and the combination of the algorithm that measures heart rate and the signal from the galvanometer results in a DHT. This would be a mobile cardiac monitor. The same would apply to accelerometers, which have algorithms to measure walking, scratching, sleeping, or tremor. Photoelectric cells, which can be used to measure blood oxygen saturation, electrochemical sensors, and a good example are continuous blood glucose monitors, which again rely on algorithms and calibration curves to make frequent measurements of blood glucose, thermocouples, which are used to measure temperature, etc. As far as these biosensors go, they can measure clinical features, some of which may be discrete events like steps, breaths, coughs, pulse beats, seizures, tremor, or forced expiratory volume. On the other hand, they may make continuous readings like continuous glucose readings, pulse oximetry, temperature measurement, ECGs, electrocardiograms, and blood pressure. It's worth reminding ourselves of some of the novel types of data that biosensors can provide. Firstly, they can provide rich continuous data instead of snapshots. So, for example, we can get the average number of steps a patient takes per day versus the 6-minute walk distance. We can get continuous glucose monitoring versus a static hemoglobin A1C, and that way we can monitor hyperglycemia and hypoglycemia and time in range and so on. DHTs give us the ability to detect rare events, for example, things like falls, arrhythmias, seizures, or apneic spells, and these often don't occur during a particular study visit. We can get data from patients who cannot respond. For example, we can measure the scratching in infants at night with atopic dermatitis. We can measure sleep in patients with dementia. We can get dose response information. For example, we can capture the on-off effects of Parkinson's drugs. We can make new types of measurements that were not possible when we saw patients in the clinic. For example, we can measure gait stability that may predict falls. We may be able to measure the amount of coughing that a patient does, sneezing, tremor. We can measure behavior patterns in dementia or in depression. And another important feature of digital health technology is that they may allow us to detect functional abnormalities very early. For example, coordination abnormalities, gait abnormalities, or reaction time. And these may be very important in, for example, neurodegenerative conditions where we want patients to be treated before their conditions advance too far. In order to rely on digital health technologies and biosensors, verification is an important process that confirms that the sensor is working as needed. And here you can see an example of a shake table that's used to test the accuracy of accelerometers to measure acceleration. On the right-hand side, you can see the performance of an accelerometer at different temperatures. An important study that's needed to give us confidence in digital health technologies is an analytic validation study. This confirms that the interpretive algorithm is working properly. And here you can see on the left-hand side the raw signal from a triaxial accelerometer, both for subjects running and subjects walking. And you can see roughly the deviations that correspond with steps. And on the right-hand side, you can see the ground truth recorded in a video recording. The video recording is scored by an observer. The interpretive algorithm gives the step count in 5 minutes at 480 with the accelerometer, and the observer reports a step count of 477 in 5 minutes. And there is just a theoretical example of how we can calculate the sensitivity and the specificity of the interpretive algorithm for measuring steps. It's important to consider confounders of the measurement as well when we rely on digital health technology. Is the result affected by how the patient wears or uses the DHT? Are there things that the patient might do that would be misinterpreted by the DHT, for example, tapping a foot or riding a bike? As part of validation, one of the questions is whether the DHT is suitable for use in the trial. This is an operational issue. Is the DHT ugly or elegant? Is it easy to put on? Is it easy to operate? Is it comfortable to wear for the time that you need to wear it? Is the battery life long enough? Is syncing data a simple process? And is it appropriate for patients to bring their own devices, for example, their own smartwatches or cell phones? Many of the DHTs that I discussed before are passive measures. They measure the patient without having the patient actually do anything other than carrying on with their normal functions. But interactive DHTs are another promising possibility for measuring clinical features or clinical performance. And here you can see examples of this. Probably in its most pedestrian form, a patient-reported outcome on a cell phone is one form of DHT. A cell phone camera can be used actively by the patients to capture a lesion. Vision tests can be done on a cell phone. Coordination tests can be done on a mobile app. This is an example of an app where there are 2 buttons on the screen and the patient has to press them alternatively as many times as they can within a given time interval. Mobile spirometers are another thing that can be used interactively. And finally, there are other more elaborate machines that can be used to measure patient strength or patient activity. You can see examples here of a portable fixed dynamometer, which is used to measure muscle strength. Now, it's important to distinguish between the tool that's used to make the measurement of a clinical feature versus a clinical endpoint that makes use of the clinical feature to evaluate a drug effect. Verification and validation that I mentioned earlier are technological assessments, and they only address how well the technology measures the clinical feature of interest. On the other hand, justification of an endpoint or clinical outcome assessment is a clinical issue. It addresses where the clinical feature is a meaningful way to assess the response to treatment, and it has nothing to do with the instrument that's used to make the measurement. This is an example of a clinical outcome assessment that was qualified by the European Medicines Agency. This is the 95th centile of the stride velocity, which was qualified as a primary endpoint in studies in ambulatory Duchenne's muscular dystrophy. What it represents is the top 5% fastest strides a patient spontaneously takes in a normal daily environment over a predefined period, say a week or whatever. And remember that this is device agnostic. It doesn't matter whether this is measured by a video camera or whether it's measured by an accelerometer. Developing endpoints that are based on digital health technology measurements, it's very important to determine that these are clinically meaningful measures of drug effect. The following are considerations when you formulate the endpoint. What exactly is being measured? In our example, this would be something like steps. What is the time window of observation? The trial may be a trial over 4 weeks. And what is the formula to calculate the response in each patient? In this example, this would be a change from week 1 to week 4 in the average daily step count. And then other considerations are whether the endpoint is a clinically meaningful measurement of drug effect, and this relies on comparison with existing benchmarks of performance. One example would be the Unified Parkinson's Disease Rating Scale in the case of patients with Parkinson's. Sometimes we may rely on other patient-reported outcomes or the 6-minute walk distance as prior benchmarks of drug activity. And it's very important to include the input from patients, from caregivers, professional societies, disease experts, and regulators when deciding whether you've established a clinically meaningful endpoint that's measured by a DHT. I included this slide just to give you an idea of the types of endpoints in general that are used in clinical trials, in registrational trials. This is a summary of those that we've seen between 2015 and 2020. And you can see that there are different types of endpoints. About 30% of trials rely on biomarkers, things like chemistry measurements, hemoglobin A1C, hematology measurements, for example, neutropenia, pathology measurements, microbiology measurements, for example, sustained virological response in HIV or plasma viral in hepatitis and plasma viral load in HIV. Imaging is another important feature often used in cancer trials, and other examples include bone mineral density, spleen volume, and then physiological or functional measurements were the endpoints in 7% of the trials we looked at. These are things like the 6-minute walk distance, which we've described, detection of normal sinus rhythm, forced expiratory volume, and sleep studies. And then 22% of our trials during that time period relied on clinical events or clinical signs, for example, death, hospitalization, major adverse cardiac events, or multiple sclerosis relapses. And finally, 32% relied on clinician-reported outcomes or patient-reported outcomes. And some of these examples are clinical scores. For example, the Toronto Western Spasmodic Torticollis was a rating scale that was used. The Hamilton Depression Rating Scale for depression, rheumatology scales, ankylosing spondylitis scales, psoriasis severity indexes, and so on. Based on that list, it's obvious that there are certain types of endpoints where mobile technology tools may play a role. They can play a role in clinical laboratory measurements, as we know, measuring things like continuous glucose monitoring, pulse oximetry. They can make physiological measurements of heart rate and rhythm, breathing and lung function, seizures, syncope, temperature, weight, sleep. They can make measures of clinical events, which I've mentioned before, seizures, falls, arrhythmias, apneic spells, and they can be used to make performance assays, for example, to assay stamina, strength, coordination, abnormal movements, and cognition. DHTs can be used in different ways in a clinical trial. They can be used for enrollment screening, and they can be used to enrich certain populations, for example, those with severe functional abnormalities. They help us quantify disease severity, and they may be able to establish the functional status at enrollment. They can be used to monitor safety, for example, the identification of rare adverse events, and they may provide real-time access to safety data. They can be used to visualize the dose effect, and they can be used for clinical trial endpoints, which we've already discussed. As far as endpoints go, the use of DHTs would be most compelling in superiority studies, where one arm of the study is shown to do better than the other arm. Our concern with non-inferiority studies is that if the DHT performs badly, it may make both arms look the same, and we may not be confident that the drug has any effect at all. Points on our regulatory position: digital health technologies and clinical trials generally do not need to be approved or cleared by FDA for marketing unless they are being used as medical devices. Our regulatory standards are the qualities of the data from the DHT adequate to provide substantial evidence of effectiveness when testing a drug? Are the data attributable to the patient? Are there processes in place to ensure data integrity, data security in transit and during storage? These are just data quality issues which our regulations address as far as electronic systems go. Brings me to my concluding slide. Electronic technology provides many new opportunities to exchange information, to obtain clinical data remotely from patients, from healthcare providers, from clinical institutions, and to make improved clinical measurements. Interoperative electronic systems will support integration of clinical care and clinical research, and these advances will play an increasing role in improving trial efficiencies, convenience for patients, and access for diverse participants, including those with rare diseases. If you're considering an innovative trial design for a drug-specific application, you should contact the relevant review division for a pre-IND meeting. For engagement on digital health technologies or decentralized clinical trial designs that are not related to a specific drug development program, you can contact us at dhts@fda.hhs.gov, and we will determine the best forum to address this. Questions? All digital health technologies for use in clinical trials need to be cleared by or approved by FDA, true or false? Well, that's false. Only those that are being used as medical devices need to be cleared or approved. Justification of an endpoint as meaningful depends on the DHT that's being used to make the measurement, true or false. That's false. Justification of an endpoint is independent of the method of measurement. And finally, trial complexity is not a concern when trials are integrated into clinical practice, true or false. And that again is false. Trial complexity is a concern. Trials integrated in clinical practice should generally be streamlined with simple procedures that align with clinical practice. And I'll end there. Thank you.
Good afternoon, everyone. My name is Meeker-O'Connell, and I head the Office of Clinical Policy, which sits within FDA's Office of the Chief Medical Officer. And today I'm going to talk to you briefly about informed consent. And I'll start with a little bit of a spoiler. It's more than just that document that a participant signs. We have about 30 minutes together, and we could probably spend hours just talking about and delving into the many different aspects of informed consent. For today, I'll walk through our general expectations for informed consent, provide insights on approaches that can improve informed consent so that it focuses on participants and the information that they need to make an informed decision. And this will include touching on what is in a draft guidance on key information in informed consent that we issued earlier this year. So let's get started. Informed consent isn't just a signature. It's not just a document. It's an ongoing dialogue. So obtaining informed consent involves disclosing relevant information to permit a potential participant to make an informed decision about whether or not that trial is the right option for them. And it's doing that in an environment or through a process that's conducive to sharing information and having discussion. Our regulations at 21 CFR Part 50 require investigators, with limited exceptions, to obtain informed consent from individuals before they can participate in clinical investigations of FDA-regulated medical products. Our regulations require consent to be in language understandable to the potential participant, and conversely, they require consent to be free of language that appears to free or release an individual or entity from liability for negligence, for fault or guilt, or what our regulations would term exculpatory language. That is, an informed consent form can't include statements like, I waive any possibility of compensation, including any right to sue for injuries that I may receive as a result of participation in this research. That would be exculpatory language that's not permitted. Finally, our regulations require that the process of obtaining an informed consent not create undue influence or cause coercion. Our regulations recognize certain groups as vulnerable to undue influence or cause coercion, but it can also be situational. For example, if a study involves a collection of additional biospecimens during surgery and you were to wait until the patient is in the OR just prior to the surgery to ask them to participate, that might create significant pressure on them. So it can also be situational. So those are general requirements, and this rule, paired with complementary efforts such as guidance, has been highly impactful. consent requirements are widely implemented, and information about studies is routinely provided to prospective participants before they enroll. And it's not just that document, as I mentioned. It's a process that begins with recruitment and extends through the end of the study. It's not just a one-and-done activity, a checkbox to be ticked that the participant has consented, we're off to the races. As just mentioned, it involves prospectively providing a potential participant with relevant information to allow them to make an informed decision, but it also assures their continued agreement and understanding throughout their participation in a trial. This means upfront that people have enough time to consider participation and they can ask questions. It can also include the researcher assessing whether or not someone understood the research. A very common form of formal assessment of understanding is the teach-back method, in which the person who's obtaining consent asks the prospective participant to state back what they understood of what was said. But again, this discussion and documentation of informed consent prior to a study is really only part of the process. During the conduct of a clinical investigation, there may be new information that arises. There could be a protocol change or new findings related to safety. And this significant new information can affect an individual's willingness to continue participating in a trial. So when this occurs, the Institutional Review Board, or IRB, needs to determine whether currently enrolled participants should be provided with the new information and given that opportunity to affirm their willingness to continue in the research. We often hear people in the research community referring to this process as reconsent, but I'll just note that our regulations don't actually use the term or define the term reconsent, although we have the concept of reaffirming willingness to continue in our regulations. Now that we've talked about general requirements, I wanted to touch on the 8 basic elements of informed consent. These 8 elements must be included in informed consent under FDA's regulations. So the idea is really to provide information to participants or to their legally authorized representative so that they can make an informed decision. We generally recommend when discussing the required elements of informed consent with prospective participants, the discussions start with the care a patient would likely receive if not part of the research, if that's relevant. And then the potential participant should be provided with information about the research. And the reason is that this sequence allows people to understand how the research differs from the care they might otherwise receive. So as you look at the basic elements on the screen, you'll find that many of these are self-explanatory, such as a clear statement that participation is voluntary, or an explanation of whom to contact for answers to pertinent questions about the research participants' rights, and who to contact in the event of a research-related injury to the participant. I wanted to highlight 2 areas in these elements where we either have seen complexity arise unnecessarily or we got frequent questions. And the first is, for the first element, it requires an explanation of the purpose of the research. And when providing that explanation, it's very tempting to go to the protocol and pluck out the language providing the scientific justification for study and insert it in the consent form. However, not the best idea. That language is often technical and complex, while the informed consent form needs to use words that are familiar to the non-medical reader, that are understandable to the participant. So that's one element. The other is actually the second element on reasonably foreseeable risks or discomforts. These need to be described in the informed consent form, including the risks or discomforts of activities required by the protocol, with a particular focus on those that carry significant risk of morbidity or mortality. And you also need to include possible risks or discomforts due to changes in a subject's medical care. So, for example, by stopping someone's current treatment and randomizing them to either an investigational drug or placebo. However, it's not necessary to describe every potential risk, especially if doing so could make the form overwhelming for participants to read. Information on risks that are more likely to occur and those that are serious should be described, because that allows prospective participants to understand the nature of the risk. Continuing with this discussion on risk, when appropriate, a statement needs to be included that a particular treatment or procedure may involve currently unforeseeable risks. This brings us to the additional elements of informed consent. These are elements that need to be included when appropriate, depending on the clinical trial. So in addition to what I just mentioned, these include the circumstances of study termination, any costs to the participant, the consequences of withdrawal, a statement that significant new findings relating to someone's willingness to continue in a study will be communicated, and lastly, the approximate number of people who will be involved in the trial. Finally, for certain trials, there is a mandatory verbatim statement that needs to be included in informed consent documents, and this is for applicable clinical trials, the subset of trials that are required to be registered into the clinicaltrials.gov databank. So what I just walked through is what's in our current effective regulations. You're probably aware that FDA published a proposed rule in September of 2022 that would, if it's finalized as proposed, would revise the content, the organization, and the presentation of information included in an informed consent. This includes proposed provisions related to key information that we believe should better facilitate important discussions between a prospective trial participant and their healthcare provider or with another trusted advisor about whether a particular clinical investigation is an appropriate option. In addition to these key information provisions, we're also proposing to add a new mandatory element of informed consent that would require a description of how information or biospecimens may be used for future research or distributed to another investigator for such future research. Finally, related to consent, we are proposing to add 3 of those additional elements of informed consent. The first is a statement about how private information or biospecimens collected during the research may be used for commercial profit, and whether the participant will or won't share in the commercial profit. Next is a statement on whether clinically relevant research results, including individual results, will be disclosed to participants, and if so, under what conditions. And finally, the third proposed additional element pertains to research involving biospecimens and would require that participants be told whether the research will or might include whole genome sequencing. So, what's in our current regulations? We just talked about what's been proposed, but if we focus only on what's in our current regulations, it represents the minimum information that needs to be provided as part of informed consent. I think it's worth noting that the consent form should also incorporate any additional information that may affect a participant's rights and welfare or willingness to participate in the research. I think another thing we lose sight of is that there's flexibility in the design of consent forms. When we talk about innovative approaches in clinical trials, I rarely hear informed consent being part of that conversation. And I think that's actually pretty unfortunate. So Christine Lee, who's the Deputy Director of our Office of Minority Health and Health Equity, has described how we often just focus on what's right in front of us. So for example, in this photo, we focus on the hands, we focus on the arms, and the elements of informed consent written on them. And we lose sight of the creative power of the in-between, the opportunity to present this information differently. As a result, we've ended up with consent documents that are too often long, complex, and legalistic, that seem more focused on the needs of sponsors or institutions than on the information needs of participants. So we think that after decades of empirical research indicating opportunities to improve, including taking full advantage of practices outlined in FDA guidance, like the use of images, the use of videos, and the appropriate use of technology, it's time for us to all increase our efforts to implement a truly participant-centered informed consent process, and further, to think how we could more meaningfully engage patients and communities as partners in the design of informed consent. At the end of the day, IRB's clinical investigators and sponsors share responsibility for making sure that consent forms and processes are adequate and meet FDA's requirements. And I think these parties should consider informed consent as much an area of innovation as other aspects of trial design and delivery. And take heart, there are existing and evolving resources that outline many approaches that can support potential participants in understanding planned research and on making an informed decision on whether to participate. So I wanted to highlight 3 final guidances on the left that may be particularly helpful. The first is our informed consent guidance from 2023, a final guidance that provides an overview of each regulatory requirement for informed consent. It also includes a Q&A section covering many of the questions we frequently get, ranging from how to appropriately consent non-English speaking participants to payments and reimbursements to trial participants. The second guidance from 2016 is specific to electronic informed consent, and most recently we published a final guidance on electronic systems, records, and signatures that can be helpful in thinking about implementation of electronic informed consent. There are also recommendations and resources from the Clinical Trials Transformation Initiative from an informed consent project they completed in 2016. This includes recommendations on both the process and the document. For example, there's a toolkit to support effective informed consent discussions. And on the document itself, CITC recommended a tiered approach where, for example, there might be a short 1 to 2 page summary of the study, followed by a section covering the regulatory elements of informed consent, and then a section with more detailed information and items that aren't part of FDA's regulations but may be required by other regulations, such as the HIPAA disclosure forms in the US. Looking at the right-hand side of the screen for ongoing work, I've already mentioned the September 2022 proposed rule, and I will shortly turn to talking a little bit about the draft key information guidance. I want to focus here on ICH E6R3. We know this is broad guidance that covers, spans everything from design to conduct, oversight, and reporting of clinical trials. And while it has a broad scope, beyond protection of research participants, it includes principles related to informed consent that I would encourage you to review. That informed consent should be fit for purpose for the trial context, that it should be clear, it should be concise, and it should be understandable. I want to touch quickly on the 2016 final guidance on electronic informed consent that highlights technological advances that could be leveraged to enhance informed consent. So this guidance really envisions an interactive informed consent that includes some of the things we've touched on: images, diagrams, graphics, and videos, and may also include embedded questions or other methods to help assess how well a potential participant has understood the information presented to them. So we recognize that some populations may prefer a more traditional paper-based informed consent, but we also recognize that that paper-based consent has limitations, especially for patients who are ill and not in a condition to read and digest lengthy documents, or for patients with low literacy, where a paper document may not be an effective communication tool. So with those considerations in mind, e-consent can provide several advantages. It gives participants options of how they want to view information, whether that's listening to it, watching a video, or reading it on the screen. And that includes the ability to tailor a screen to their needs. For example, font size. I've been on many Zoom calls over the past 4 or 5 years, and there's always someone, it's often me, asking the presenter to make the text just a little bit bigger. These e-consent approaches put that control in the hands of the potential participant. They also permit things like hyperlinks to supplemental information that can go into greater detail if desired. And importantly, e-consent facilitates remote consent, being a patient or participant being able to walk through the information at their own pace in a location that's their preference. So they don't have to necessarily travel to a particular trial site to complete an informed consent for the study. And that is an important decentralized element that supports efforts to expand the tent of research. So we've come to our first challenge question, and I would ask you, which of the following statements is not true? Is it FDA regulations include 8 elements that must be included in all informed consent documents? Is it not true that informed consent is an ongoing process? Is it that FDA regulations do not permit the use of images, graphics, or cartoons in informed consent? Or is it that informed consent cannot contain exculpatory language? I really hope you picked this one. C, that it is incorrect that our regulations don't allow the use of technology or cartoons or other visuals. I've heard that there are some people who are nervous about using graphics or images, and we've heard directly from patients that this can really help them digest often complex information, and there is definitely not a regulatory prohibition. Now let me move to the last topic of the day for me, talking a little bit about the key information provisions that are in the proposed rule that I mentioned, as well as about a related draft guidance that we issued earlier this year. So the key information provisions, there are 2 of them. One is that consent needs to begin with key information. And second, that the whole consent be organized and presented in a way that facilitates understanding. So those provisions are discussed in more detail in the guidance, the draft guidance that I mentioned, which talks about how to assess which elements of consent may be key information for a particular study in the populations that are intended to be enrolled. The guidance also recommends that sponsors consult with patient groups and with communities to help identify what information is most relevant to them. That is, what information would be key and should be included up front. Finally, in the spirit of being clear and concise, this guidance notes that key information in limited circumstances could actually form the entire consent if it includes all the required basic elements and sufficient information. So, for example, for a very simple minimal risk trial, the key information could be the entirety of the consent form. You might be wondering, Anne, stop a second. What is key information? It's the specific information or subset of information that is most likely to assist a prospective participant or their legally authorized representative in understanding the reasons why they might or might not want to participate in the research. It is concise, it is focused, organized in a way to promote understanding, and it's the first thing presented to a potential participant under the provisions in our proposed rule. Now, thinking about this key information, there are many ways that you could conceive of designing this section or this information and presenting it to participants to help them understand the study. The working group at FDA that developed the draft guidance created one example of how you might design the key information section of the consent using different tools and tips of approaches that can help aid understanding. This slide shows the example from the end of the guidance. And for example here, don't focus on the words themselves. We'll walk through the approaches used to organize and display the information to help the participants understand the trial. But I will emphasize that this is just one approach. Again, there are many different ways that you could conceive of presenting this key information to help facilitate participant understanding. So in the guidance or draft guidance, we recommend the following tools or aids that can be used to improve the key information in informed consent. One is the bubble format. So you'll see information grouped into distinct chunks, which may be easier to understand. There's also bullet points to simplify the text. The bulleted information included in those rounded boxes creates a small unit of text that becomes easier for somebody reading it to digest. There are areas of white space or empty space around the boxes, just a good design principle. This is an example that's limited to a few pages. It uses simple text where possible, and it adds hyperlinks to where an individual could find more information in the entirety of the consent form. So this is an example of how applying design tools together can help make information more available and easier to understand. And starting consent forms with the key information that similar to what you might see here will be a requirement when our rule is finalized. The kind of example of how to organize key information in our draft guidance is a recommendation. It is not a requirement. And again, you can be creative and use other approaches. So I talked about key information being two provisions. We just talked to the first. This is the second. And important here is the concept that the informed consent as a whole, we need to up our game with it. It needs to be organized, presented in a way that facilitates understanding and decision making. This is where what we talked about earlier comes in. Be creative. Think of informed consent as an area of innovation. Leverage technology where appropriate. Use video, use graphics, even use cartoons. And when I talked about using technology where appropriate, that to me is because engaging and getting feedback from potential research participants and communities will give important insights into how these parties prefer to receive information. But all of these approaches can be helpful in improving the understanding of participants when they are presented with the complex information in consent. So this brings us to our final challenge question of the day. Which one of these is true? Is it A, our proposed key information provisions include a very specific format for presenting key information? This may be tempting, but no. Key information needs to be presented first, but as we discussed, there are many approaches that can be used to structure, organize key information. Our draft guidance provides just one example. Is it B? Do our proposed regulations require key information to be the last item reviewed? No, as I just mentioned, it's the opposite. Key information, the information most relevant to decision making, comes first. Maybe is it C, that key information is intended to replace consent? Now this could be tricky. While there may be limited cases in which key information for a simple trial contains all the required elements and sufficient information that it could be the entire consent form, key information generally isn't intended to replace informed consent, nor does it replace the dialogue that occurs as part of the consent process. What it's intended to do, which you know if you selected D, is to facilitate understanding about why an individual might or might not want to participate in a trial. So that brings me to the end with a few take-homes. Be concise, be clear, and use plain language in your consent forms. Really engage those who may be potential participants in the trial and the communities they come from. They will give you keen insights into informed consent content and the process. Leverage innovations where you can, and whether it's with the content or approaches to delivering informed consent. And remember, our regulations don't prohibit creativity. We know we've talked for a while, for many years, about improving consent and moving away from 40-page documents that are single-spaced text. That's not going to change unless all of us that are interested in the consent process or have obligations in it actually make those changes. So I would encourage you to apply some of these approaches then with the next consent form you design. And that's it for me. I look forward to the discussion and your questions. Thank you so much.
Hi, everyone. I'm back. Let me begin by thanking Leonard and Anne for their insightful presentations. This session covered the topic of decentralized clinical trials and the ways in which digital health technologies are creating new clinical trial opportunities. Furthermore, Anne covered in detail the general requirements for informed consent, elements of informed consent, and what constitutes the informed consent process. With that recap, let's go ahead and dive into some questions from the audience, and we have a lot of questions. The first one is for you, Leonard. This is multiple questions, so bear with me, please. How is a clinical investigator expected to exercise oversight of home visit staff, mobile phlebotomy or nursing services? Would they be expected to verify GCP HASP training? Ensure these providers understand their obligation to report adverse events? An example of this is a large hematoma as a result of a blood draw might not be recognized by a phlebotomist as a reportable event due to clinical trial requirements. So what happens in this kind of a situation?
Thanks very much. A good question, which really gets to the heart of decentralized trials. So I think the first thing that's worth realizing is that adverse events don't just happen at the times that patients go for their traditional trial visits during the half hour they're with investigators. They happen anytime. And in these sorts of trials, in traditional trials, there's no expectation that the investigator will witness everything that happens. So I think the same approach is being taken with decentralized clinical trials. When it's reasonable, investigators should supervise certain things where the risk is anticipated. and that can be done by video supervision, or as appropriate, a hybrid approach can be taken, and areas where there's potential risk or concern can be done at a trial site. Another point that I thought is probably worth making is that even in traditional clinical trials, patients are often sent away for procedures. They may go for a scan with contrast, and individuals involved in those procedures are not part of the trial. So I don't think it would be expected that they would report adverse events. I think the bottom line is that, as for traditional trials, I think decentralized trials have to have protocols that very clearly have procedures for making sure that adverse events are captured. And this would be the usual way in trial visits. Investigators would be expected to ask about adverse events. If there were very specific concerns, I think investigators would prompt patients for that sort of information. There was another part to the question about checking the qualifications of healthcare providers. And I think there we're deferring to qualified individuals in the clinical care environment. And the qualifications for doing a phlebotomy in a clinical care environment as opposed to a research environment would really be no different. And many procedures are covered by both the clinical care and the research environment. So as long as these are qualified people, they're licensed, I don't believe that there would be any extra need to check their qualifications or competencies. Thanks for the question.
Thanks, Leonard. That was helpful. And I'll move on to you. Do you have any recommendations on tools we can use to make sure the text of an informed consent form is understandable to people who don't have medical knowledge?
Yeah, and that's a great question. I think, I would encourage engaging kind of the institutional review board or ethics committee to see kind of whether they may have resources. There are many websites with information about health literacy, but to my mind, one of the best ways to make sure that the language is understandable to kind of the potential participants, the populations that may be enrolled, is to actually test the consent form, get feedback on it from the communities who may be engaged. This is something, if you look at the updated, recently released Declaration of Helsinki, talks about having participants and communities engaged early. It seems like looking at whether the informed consent form is understandable would be an excellent way to do that. So thanks so much for the question.
Thanks, Anne. Leonard, coming back to you. While telemedicine has done a lot to expand what can be done in clinical trials, there also seems to be a trend towards removing the federal policies that support telemedicine and going back to the time when people would have to go to a physical clinic for care or to participate in clinical research. So the question is, should researchers be concerned that conducting remote trials may become more difficult as telemedicine-related policies are rolled back? What are your thoughts on that one?
It's an interesting question. I think there's a clear distinction between telemedicine in the practice of medicine and telemedicine as part of research. Obviously, clinical care is a different sort of activity, and I'm not aware of these efforts to roll back telemedicine, but I mean, if they do exist, they're related to clinical care. In the realm of clinical research, the degree of remote interactions with patients would be driven by a protocol. They would have to be, it would be essential that those procedures that were going to be done remotely were considered as being suitable for remote performance. If these are just telephone follow-ups or video follow-ups, this is nothing new. This has been done in clinical trials for years. So I think a lot of the remote procedures that are specific to clinical trials really aren't part of the clinical care environment, where you may need to have a more hands-on approach. But again, I think one has to be judicious about what one decides to do remotely in a clinical trial and what needs to be done at a clinical site. Thanks for the question.
Thanks, Leonard. That's helpful. And not letting you go yet, I do have a follow-up question. With the very high prevalence of mistrust, distrust for medical research in America, what realistic chance of successful implementation do this decentralized and point-of-care trials have? Leonard?
I would hope that they have a greater chance of success than traditional trials for a couple of reasons. First of all, with trials which are integrated into the healthcare system, the big advantage is that patients will be using their usual healthcare providers. And I think that's a very important feature in improving trust in general. And certainly in my clinical experience, patients often come to me to ask whether participation in a trial is a reasonable idea. And I think it certainly gives patients a lot more comfort when they're involved with people they know. As far as decentralized procedures go in general, I don't think there's much of a difference between that and going to a clinical trial site in a traditional trial. I think the advantage of decentralized procedures is really just convenience, making it easy for patients to participate. I'm not sure whether the decentralized trials as opposed to clinical trials have as opposed to site-based trials have a different impact on patients' trust. I would think it probably is much the same, but perhaps if decentralized trials can incorporate culturally suitable trial personnel and accommodate differences in languages, that may give patients more comfort. So I think there are a couple of reasons to be optimistic. Thanks for the question.
That makes sense, Leonard. Continuing to be optimistic here. And moving on to you, we do have, we need some advice from you about how to resolve a situation. The situation is, if the subject's visits to the site are rare and several informed consent forms were issued in between, would it be correct to sign them all or just the latest one, as the latest version contains all previously available information? And
so I think this question is probably going to be answered with the classic "it depends." So I think what I think is important to tease out here is that we talked about in the presentation that there may be significant new information, whether it's new findings related to safety or a change in the protocol, that might actually impact someone's willingness to remain in a clinical trial. And the IRB is in the role of determining whether currently enrolled subjects need to be provided with the new information and given the opportunity to affirm their willingness to continue in the research. And so, to my mind, what the IRB is looking at, whether also often is advising on whether the investigator should provide currently enrolled participants with the new information, either with the revised consent form or through an alternative method. And my question about having multiple consent forms that have been issued between study visits is that, if somebody's willingness to continue to participate may be impacted by the information, I think to my mind, it's less about which version do we sign when they finally come to the clinic, but how do we make sure that information gets to them so that they can weigh it. And that seems like an ideal opportunity to leverage some of the practices like electronic informed consent or remote informed consent rather than waiting for when somebody comes into a clinic visit. So thanks for the question.
Thanks, Anne. That's helpful. And if you could stay on for another couple of minutes, I do have a question. It's a very interesting question. How would you suggest making sure that individuals from a culture that are reluctant to question authority have fully understood what they are consenting to and are not just signing because they feel they're expected to? Any thoughts on that, Anne?
What a great question. So I think I have a couple thoughts. One relates to the consent process itself. And how the person who is conducting that consent discussion engages with the participants. So we've had the opportunity to engage with, I think I mentioned our REACH consortium and through our Office of Minority Health and Health Equity. And some of the practices there seem on point. So instead of conducting the discussion, conduct the discussion seated at kind of the same level as the subject. So, not standing over them, in a position that might create an impression of of a power differential. I think having family members or others present. So that's there are some considerations for thinking about the process and that discussion. The other kind of easy to implement approach is simply to, when the discussion has concluded, to ask a simple question like, I know I've just explained a lot of information to you and given a lot of material. Can you, basically asking something along the lines of, just to make sure that I've, explained clearly, can you let me know kind of what you heard? So it becomes more of a conversation. So those are just a few thoughts, but again, excellent question, and thanks for the opportunity to respond.
Thank you so much, Anne. Leonard, I have one for you here. Is the frequency of validation or calibration of equipments or instruments considered in the DHT guidances?
Thanks for the question there, Millie. So to some extent it is. Validation, at least analytic validation for digital health technologies that use interpretive algorithms, is something that has to be sort of finalized before the trial goes on. We have to be confident that the DHT is making a reliable measurement. Now, having said that, it's possible that in some situations there may be improvement of the interpretive algorithm. In general, it's not a good idea to change algorithms post initiation of the trial. That can obviously lead to all sorts of interpretive problems and may require separate analyses for those who used the device before and after it was modified. So I think as far as validation may need to be repeated if there was some sort of change or an update to the DHT, but in general we would prefer those things to be locked. Yeah, I think, Millie, can you remind me of the second part of the question there? I think it was.
No, that was it, but whether the frequency of validation or calibration of equipments considered in DHT guidances.
Sorry, apologies. You got the other question was about calibration, and I think calibration depends on the DHT. If we're thinking of a continuous glucose monitor or something like that, calibration would be something that would be part of the DHT instructions for use. So I don't think that would be a specific trial consideration. Thanks for that.
Thanks, Leonard. We only have a minute left, so I'm going to squeeze in a question here, Leonard, for you. What is the regulatory position of using medical devices in decentralized and real-world evidence trials? Are you aware of any further considerations to be made?
I think in the guidance we mentioned that using medical devices which are approved or cleared has an advantage because obviously they've been vetted, the measurements are reliable, we have the information. And so in general, when suitable devices, approved medical devices are available, we would encourage their use, and sponsors using those devices can often refer to existing information on their validation, their usability studies, and so on and so forth. So certainly are advantages, but there are many situations where we anticipate that DHTs will not be medical devices at all. They may be wellness devices, things that measure steps or sleep or various other sort of patient reported outcomes. And in these situations, I guess we would evaluate each of those DHTs on their own merits. So in short, advantages in DHTs which have been approved when appropriate for the trial, but certainly open to all sorts of other DHTs, provided the suitable information that we need is provided. Thanks.
Thank you so much. Well, thank you once again, Leonard and Anne, for your presentations and for taking the time to answer the questions from our participants. Thank you to our audience for your interest and participation. I'm very thankful to have had the opportunity to moderate this engaging session. I would now like to hand over to Leonard for the next session. Thank you, everyone.
Well, thank you very much, Millie, for your excellent moderation. And thanks to Anne for all her very helpful comments on informed consent. So this brings us to the last session of the day. I guess the aim of this session is to showcase breakthroughs in medical science. And for this particular session, we've chosen two areas, mRNA vaccines and gene therapy. These are biological innovations with tremendous promise for a lot of diseases that previously were regarded as untreatable or difficult to manage, but they do raise unique considerations, not least of all for regulators. So I think we're all very excited to hear, first of all, from Karen Bock about mRNA vaccines, and she'll be followed by Nicole Verdun, who'll be telling us about innovative therapeutics and gene therapy. So Karen, over to you.
Good afternoon. Thank you for attending this presentation. My name is Karin Bock. I am the Deputy Office Director of the Office of Vaccines Research and Review here at CITC FDA. And today I wanted to talk to you about mRNA vaccines. This is my disclaimer. My comments are informal and do not bind or obligate FDA, and I do not have any disclosures or conflicts of interest to declare. So at a glance, I wanted to give you a map of what the presentation will look like. We'll have a brief introduction to the topic. I'm going to tell you a little bit about mRNA vaccines technology, mRNA vaccines efficacy, which a lot of the data is from the SARS-CoV-2 vaccines, mRNA vaccines safety in general, also from vaccines we've been using for a while, like the SARS-CoV-2, and also biodistribution of mRNA in general. We're also going to talk about what we're still doing in terms of formula updates for vaccines, and how is the durability of the vaccines we're using via mRNA. Since the pandemic, there's been other approved mRNA vaccines, and we'll describe those. And of course, there's many of them that are in different stages of development. I'll tell you a few summary and conclusions that I think will be useful after listening to this. And of course, we have a couple of challenge questions for your entertainment. Okay, so we've been using vaccines for a couple of centuries now, but really the scientific advance on developing new vaccines and new technologies for vaccines has been in the last 70 years. Starting, of course, since we figured out how to grow viruses in vitro, and because of that, we were able to produce vaccines at larger scale. But since the discovery of the polio vaccine, we've made several advances. We figured out how to make recombinant vaccines like hepatitis B. We've had live attenuated vaccines like rotavirus. We have virus-like particles vaccines that have been very, very successful, like HPV. And of course, in 2020, we approved for the first time a new technology for vaccines based on mRNA. But even though mRNA is the new shiny object in our portfolio, and it's been very successful so far, we still have a lot of very advanced vaccine platforms and technologies that we're still using today. and we couldn't live without because different pathogens and different targets necessitate different platforms and different approaches. So we have inactivated viruses, we have viral-like particles, and live attenuated viruses, and we have other kinds of platforms, or what we define as platforms, like viral vector-based vaccines that we also use not only in general but also for emergency situations. So what is the technology behind mRNA vaccines? So we've been able to figure out how to use mRNA inserted into a cell and translate a protein for a while, since the 90s. But there had to be several breakthroughs for us to come from that discovery all the way to today, using mRNA as a tool for vaccination. There are several breakthroughs that are on this slide, and I'm going to describe to you. One of them was figuring out that we had to cap the mRNA. If you worked in a lab, you know that mRNA is very sensitive and it degrades very quickly. So capping helped with that. Tailing also helped with that, and also improving the translation efficiency of the gene of interest in an mRNA vaccine or molecule. We also use codon optimization in order to improve translation efficiency. And most importantly, we had to figure out how to get the mRNA molecule from the syringe all the way into your cells. And one of the most effective technologies to do that is through lipid nanoparticles. mRNA is also very reactogenic and can greatly stimulate the innate immune response. So scientists figure out that replacing the typical uridine of the RNA with pseudouridine, which is a modified molecule of uridine, reduces the reactogenicity by not stimulating the innate immune response so much. But even if you figure out all the RNA technology to have an mRNA vaccine, you still have the typical development and discovery issues that you have with any vaccine. You have to determine which one is going to be your antigen based on the pathogen that is your target. And if that antigen is going to be native and you're going to use it as is in the virus or in the bacteria, or you're going to modify it or add any other design to your vaccine. So this gives us a typical mRNA construct overview where you have your gene of interest in the middle. That's exactly what you want your mRNA to express, and that will be your antigen, your immunogen. But you also have to have an untranslated region in the 3'. You have to have a cap in the 5', poly A tail, a UTR, and all this. And of course, like we said before, the pseudouridines instead of uridines. So all this enables the mRNA construct to be a successful vaccine. Manufacturing of mRNA vaccines is atypical from other vaccines we've seen so far, mostly because we consider this manufacturing technology to be synthetic, not use biological materials. We still use sometimes biological materials to develop the initial plasmid that we need in order to eventually synthesize the mRNA. But besides that, the whole process doesn't require biological materials like cell culture, fetal bovine serum, or any other biological material, and it's done in a synthetic way. So you go from the sequence design to the in vitro transcription process, and then it's very important that you purify the unused reagents in your in vitro transcription process to have the pure mRNA molecule that is going to be your vaccine. And of course, then you have to have a method, a technology, in order to combine your mRNA vaccine with your lipid nanoparticle, and then filter the product before it gets formulated into a vial. So what happens once an individual gets injected with an mRNA vaccine. So mRNA vaccines can be transfected into muscle cells, and those are the cells that mostly translate the antigen of interest, but they can also be translated into antigen-presenting cells, which also is useful to activate the immune response. Of course, these antigens are transported to the lymph nodes, and they have lymph node resident cells that initiate the cascade of the immune response. But the muscle cells are the main recipients of your mRNA. So as we were saying before, it is important that you have modified uridines and you also have purified final mRNA product, because the innate immune response is very sensitive to any non-purified mRNA molecule, and you can have a very reactogenic vaccine, as you see here on the left, when a lot of the toll-like receptors and cytokines are activated when you inject an unpurified mRNA. And you, on the right, you see that if you use a well-constructed and purified vaccine, you let the adaptive immune response be the main consequence of that injection. mRNA vaccines have many advantages, like we discussed before. They are synthetic in manufacturing, they are faster to produce, they stimulate the innate immune response, but they also have other advantages because the antigen is translated inside the cell, meaning the antigen is delivered intracellularly. So they're not only able to produce antibodies and CD4 and stimulate the CD4 cells, they also stimulate CD8 cells. And that gives another branch of the immune response activated and ready to protect against an infection or disease. So the pros are that this is rapidly translated, vaccination is very fast, the RNA can be, of course, modified, and there is no prior immunity that can damp the immunization process, as in the case of other platforms like adenoviral vectors. You can sometimes have a vector where the individual that is being immunized has prior immunity, and that's going to lower the immunity level. And the last example we have here is a typical protein in adjuvant vaccine. While they're very useful for certain cases, the disadvantage of these approaches are that they don't have CD8 type immune response. So we have several overall advantages of mRNA technology for vaccines. Is a platform production, meaning that you can establish a method of production of the vaccine, and then by changing the gene of interest, you can use that same method of production for many different types of vaccines, which is not possible with other technologies. In general, it's biologics or cell-free production, which it's less complicated in terms of releasing or assays that you have to make sure you do in order to check the quality and the potency of that vaccine. There is a speed of manufacturing, again, because it is a synthetic and in vitro process, is much more is faster to scale and to manufacture in general. There is also faster speed of release because there are less assays required for release, just because of the cell-free production and biologic-free production. And in some cases, compared to some of other vaccines, the immune response might be more sophisticated, although this is not necessarily ideal for every pathogen you're going to have to work with. So this is just one of the advantages depending on the pathogen of interest. So what is the efficacy of mRNA vaccines? So during the pandemic, the US had an Operation Warp Speed, to develop many types of vaccines. they attempted to develop 2 mRNA vaccines from Moderna and Pfizer. And those vaccines not only were mRNA-based, they also had a specially designed antigen that was delivered intracellularly. So both of those approaches contribute to the final efficacy of a vaccine. But initially, when the original SARS-CoV-2 virus was circulating during 2020, and we were able to study in a large phase 3 trial the efficacy of these vaccines, mRNA vaccines proved to be very efficacious. In this case, you have both Pfizer and Moderna's initial results, and you can see that both are more than 90% efficacious, and even more for severe disease. And this was considered a huge success. It still will be considered a huge success for any vaccine that we study. And for instance, compared to the adenovirus-based vaccine, that was a different platform also being developed at the same time, which was slightly less efficacious with 72%. And not only that, you can see in these Kaplan curves how really the control and vaccinated groups are separated early, even a little bit over a week after vaccination. And it's very clear that vaccination is very, very effective against disease, and especially, like I said before, against severe disease. And in the case of the vector-based vaccine, the efficacy was a little bit lower, and the control group was not as separated from the vaccination. But overall, any of these results, 90% efficacy, 70% efficacy, it's considered a very successful vaccination. So we also continue to study vaccine efficacy and effectiveness, not only during the original approval of mRNA vaccines, but eventually when we had boosters, and we were able to observe that if a booster is well matched with the circulating virus, the vaccine continues to be very efficacious at preventing disease. We have, as I said, other vaccines in the market. The first one is a RSV-based mRNA vaccine, and this vaccine has also proven in a large phase 3 trial, and eventually was approved with this trial as a base of evidence that was also very effective at preventing RSV disease, and the efficacy is also over 90%. So in general, not only talking about the trials that were used to approve or authorize these vaccines, we also followed vaccine effectiveness once the vaccine was used in the US population. And as you can see here, the difference between the rates of disease in vaccinated and unvaccinated is really outstanding, especially during the early late 2021 when the virus was circulating. So these vaccines are very effective and have been proven to be very useful in the field as well, not only in the trials. So since we know that vaccines are really being effective in the field, I wanted to also talk about the other parts that we study when we evaluate a vaccine, which is safety, and to evaluate the benefit risk ratio of a vaccine. So what is, first of all, the biodistribution of mRNA once it gets injected into the arm? Of course, it's important to highlight, even though I'm sure you all know this, that mRNA does not travel to the cell nucleus as opposed to DNA. mRNA is biologically always present in the cytoplasm, and it doesn't travel to the nucleus, which is why it's impossible to get incorporated into our DNA. The antigen, however, can be detected in blood as soon as 24 hours after vaccination, and but it's rarely detected after the second dose of vaccinations. Some studies have shown that the antigen, in this case the spike for SARS-CoV-2, It's also detectable in germinal centers in the lymph node for a long time, 120 days post-vaccination. So while you might not be able to detect it in blood, it is usual for antigens to stay for a long time in germinal centers. mRNA, in general, the vaccine itself is found in blood within hours and for approximately a month after vaccination. And the mRNA LNP, which is the technology we use to get the mRNA inside the cell, is detected at the injection site and is processed by the liver. So it's also detected by the liver. And LNPs, both LNPs, mRNAs, and also the antigen can be detected in various organs and sometimes in breast milk or other tissues. And the biodistribution in general seems to be correlated with different types of LNP formulation. So safety has always been a priority when developing any vaccine, and it was certainly a priority in the middle of a pandemic when developing the SARS-CoV-2 vaccines. Although we, as you can see on this slide, we took only 11 months to develop and authorize a COVID-19 vaccine compared to other vaccines that took years and years to be designed, developed, and approved. Safety was still a priority, and it was not compromised in the development of the vaccine. And why can I say this? Because even though it was less than a year to develop that vaccine, the work that contributed to the development of that vaccine had started at least 10 years before. And the development of the vaccine was done not only by one sponsor, but by in this case for the Moderna vaccine, by a group of very experienced scientists and vaccinologists that contributed to the speed of development of that vaccine. But the work that had been happening 10 years at least before the vaccine started to be developed was the work that helped us understand what kind of antigen we needed to use, how the antigen was going to be modified to be most effective, and how we could incorporate it into an mRNA vaccine and have a successful vaccination. So all that work had been already been done with other coronaviruses and with other viruses. So we were very well prepared for the very, very rapid development of the SARS-CoV-2 vaccine. And safety was also a priority because we know that there are cases, especially with respiratory viruses, of vaccines that have been designed in a very basic way, and they can cause what we call vaccine-enhanced disease. So we did have examples of very basic vaccines, in this case, formaldehyde inactivated vaccine that had a severe safety signal. So we were very careful, and we had all the scientific tools and knowledge in order to try to ensure as much as we could the safety of that vaccine. So the United States governments took risks developing those vaccines fast. Mostly those risks were financial. We did a lot of things at the same time without knowing whether the vaccine was going to be successful, especially manufacturing a lot of vaccine without knowing if that vaccine had even been successful in a phase 3 trial. But we did not take safety risks or other scientific risks. We use standard phase 1 and phase 3 protocols, and the vaccine follow-up on safety was ongoing. It was a little bit shorter by the time it was authorized, but it kept being monitored after that. So what is the benefit risk analysis of a vaccine, and how do we think about this in the case of mRNA? So there are certain safety events that have been described for the mRNA SARS-CoV-2 vaccine for both Pfizer and Moderna. And in this case, it's myocarditis and pericarditis is one of those examples. And you can see here that there is some elevated risk after vaccination, especially before 10 days after vaccination of pericarditis and myocarditis. And yes, every intervention, every vaccine, every drug is going to have some risk, and that's why we always assess the benefit over the risk of vaccinating an individual. But we carefully understand and survey any kind of safety event, in this case myocarditis and pericarditis, and inform the public of those safety events. It's also important to keep in mind, especially during this benefit risk analysis, that some of these safety events are also part of the original infection, the virus infection. And as you can see on this CDC warning, that the heart complications are higher after COVID infection than after any mRNA vaccination. So you always have to keep in mind both what are the benefits and what are the risks of vaccination. And that's what we do at FDA. So what is mRNA has been described as maybe not as durable as other platforms. So what is the mRNA vaccine durability, and how do we work to have formula updates? So is the immunity to mRNA express antigens durable? I think that question depends both on the mRNA vaccine and also on the kind of antigen you're trying to insert. If the natural infection is not durable, it's very unlikely that the vaccine durability is going to be more durable than the natural infection. So we know that mRNA vaccines produce antibodies that last about 6 months. As you can see, it starts declining at lower levels at 6 months. We also know that we have memory cells, and that we will likely have to rely on those memory cells and memory T cells to recall the immune response if we get re-exposed to the virus. But in terms of antibodies, yes, the immune response declines, and you will likely have to be either reinfected or revaccinated in order to increase that antibody immune response, but you still have memory B cells and memory T cells that are able to be recalled. So how we approach updating vaccine composition. This is a similar process as we do with influenza vaccines. We backtrack the vaccine campaign and how much time we need to give manufacturers and sponsors in order to effectively manufacture the selected strain or the selected new booster. And we also have a clear list of requirements. What is the CMC data, the non-clinical data, and the immunogenicity data that they will need to provide in order for a new booster vaccine to be approved once the advisory committee makes the recommendation on the composition decision. So all those campaigns are followed every year and optimized every year. And we anticipate that this process, the process we use for influenza, will also apply in general to COVID-19 vaccines moving forward. So what is our role in general for COVID vaccines, including mRNA vaccines? We do have an advisory committee that does strain selection, and we have reference standard production. We do work to have lot release ready for seasonal vaccination. We evaluate safety and efficacy, and we help with post-market surveillance and advancing vaccine technology. And one of the most important things we do is helping to ensure public confidence by being very thorough when evaluating safety and efficacy and analyzing the benefit-risk ratio. So the last thing I wanted to talk to you about is the future of mRNA vaccines. We have approved mRNA vaccines like SARS-CoV-2 and RSV, but we have many others that are being developed. So we have examples for cytomegalovirus. That's one that is pretty advanced. We are close to also the end of development for influenza virus mRNA vaccines, HIV, Zika, rabies, and VZV, among others. This is just a summary example of all the, in this case, viral vaccines that are being developed with mRNA technology. So my summary and conclusions are mRNA technology was being developed for decades before the emergence of SARS-CoV-2. That's why we were able to use it so fast and so successfully. mRNA is an effective method to deliver an antigen. Careful vaccine design and antigen consideration is still needed, like for any vaccine. mRNA vaccines have been demonstrated to be safe and effective against COVID-19, and many other mRNA vaccines are in the pipeline, including the recently approved RSV mRNA vaccine. The ability of the mRNA platform to induce durable immunity is incompletely understood and still being studied. The mRNA platform advantages and cell-free manufacturing make it an ideal system for not only rapid response against emergencies, but also, of course, for the yearly updates of COVID-19 vaccines. So my challenge questions are, mRNA vaccines are the newest and most effective vaccines on the market. Do you think that statement is true? It is not. There are many technologies that are effective, and they have different advantages and disadvantages that might work better for your design depending on your target pathogen. mRNA vaccines have the advantage of eliciting CD8 positive immune responses through intracellular delivery of the target antigen. This is true. This is one of the advantages of delivering an immunogen intracellularly, just like the virus itself does it and other platforms do it. So having another part of the immune system stimulated is also helpful. Thank you very much. I hope you enjoyed the presentation, and looking forward to the questions. Bye.
Hello, my name is Nicole Verdun, and I'm a Super Office Director in the Office of Therapeutic Products in the Center for Biologics Evaluation and Research here at FDA. And today I'm here to talk to you about innovative therapeutics, gene therapy. The mission of the Center for Biologics Evaluation and Research is to ensure the safety, purity, potency, and effectiveness of biological products, including vaccines, allergenics, blood and blood products, cells, tissues, and gene therapies for the prevention, diagnosis, and treatment of human diseases, conditions, or injury. Through CBER's mission, the center also seeks to protect the public against the threats of emerging infectious diseases and bioterrorism. The Super Office of Therapeutic Products was established in March of 2023 in the Center for Biologics Evaluation and Research, with the goal of creating flexibility and capacity for future growth, and to improve discipline alignment, increase review capacity, and enhance expertise in both cell and gene therapy. The Super Office of Therapeutic Products is made up of 6 offices. The Office of Gene Therapy, Chemistry, Manufacturing, and Control, or CMC, the Office of Cellular Therapy and Human Tissue, CMC, Office of Plasma Protein Therapeutics, CMC, the Office of Clinical Evaluation, the Office of Pharmacology Toxicology, and the Office of Review Management and Regulatory Review. OTP regulates a diverse array of products, including gene therapies. The gene therapies that we regulate include ex vivo, genetically modified cells, non-viral vectors such as plasmids, replication-deficient viral vectors such as adenovirus, adeno-associated virus, or lentivirus, replication-competent viral vectors such as measles, adenovirus, and vaccinia, and microbial vectors, such as Listeria and Salmonella. In addition, we regulate stem cell and stem cell-derived products, terminally differentiated cell therapies, therapeutic vaccines, and other antigen-specific active immunotherapies, such as cancer vaccines and immunotherapies such as dendritic cells, lymphocyte-based therapies, cancer cell-based therapies, peptides, proteins, and non-infectious disease therapeutic vaccines, such as peptides, proteins, and small molecules. In addition, we regulate blood and plasma-derived products, such as purified and recombinant proteins for hematology or coagulation factors, xenotransplantation, and in the past year, we have approved both a pig heart and kidney for transplantation, human tissues, and also some devices. Now I'm going to talk a little bit about gene therapy product development. Gene therapies can be delivered via either ex vivo or in vivo administration. With in vivo administration, on the right side, the gene therapy product is administered directly to the subject. As an example, here you can see an adeno-associated virus, vector, or lipid nanoparticle encapsulating nucleic acid, such as messenger RNA. These vectors could be administered intravenously, where they could travel to the liver perhaps and correct a monogenic disorder. In ex vivo administration, cells are removed from the patient and genetically modified in culture outside the body with vectors such as lentivirus or naked DNA. The genetically modified cells are then reintroduced into the patient. One example of this is CAR T cells for cancer therapies. There is a diverse array of gene therapy products. The figure here shows the 2022 database of the different vectors used in gene therapy clinical trials. A large portion includes replication-deficient viral vectors, such as adenovirus, AAV, retroviruses, and lentiviral vectors. Other vectors include non-viral vectors, such as plasmid DNA, mRNA. In addition, you have ex vivo genetically modified cells and replication-competent viruses and microbial vectors. Interactions with us occur at various stages of product development, starting with interact meetings, which could be used for early discussion on CMC, pharm tox, or clinical related issues. Interact stands for Initial Targeted Engagement for Regulatory Advice on CBER or CDER products. And you can, at the time of these interact requests, have preliminary evidence or proof of concept studies and really want to engage with the FDA early. In addition, you see we have several different phases of meetings, including at the pre-IND phase, end of phase meetings, pre-BLA meetings, mid-cycle and late cycle meetings. In addition, we have an entire set of meetings that you can request that are discipline specific, and we welcome all communication with sponsors. Key differences in gene therapy development are, unlike other medical products, gene therapies often involve new technologies to manufacture the product, sometimes invasive procedures to administer the treatment, potential long-lasting effects, which makes it different than drugs, and potential risk of unintended changes in DNA or rejection by the body. So all of these are careful regulatory considerations when we are discussing products with companies and with academia. Considerations for chemistry, manufacturing, and control of gene therapy products in development can be quite complex. In particular, in early phases, it's important to have characterization of your product, understand the mechanism of action as much as possible. Considerations of the cellular kinetics and vector biodistribution are also needed. And in later phases, quality and development of potency assays is quite important, but it is equally important to consider these things in early phases of your product lifecycle. Major manufacturing changes can also require demonstration of product comparability. Prolonged biological activity and the need for long-term follow-up are particular considerations for gene therapy products, and the procedures for product delivery, including product device compatibility, monitoring for immunogenicity during development, and monitoring for off-target effects, including tumorigenicity and insertional mutagenesis. Particular considerations for preclinical development include the development of animal models in both healthy and disease-related states, depending on the product. And scientific justification for model selection is needed. In addition, comparative physiology and tissue type helps with extrapolation to clinical dose levels. Product kinetic profiles, such as the cell fate or vector biodistribution, the route of administration should be as close as possible to the clinical scenario in the preclinical development phase. The timing and rate of administration, anatomical location, activity of the product, and the local microenvironment are all considerations in the development of your preclinical program. In addition, standard toxicology assessments such as mortality, laboratory observations on treatment, body weights, gross and histopathology, and other endpoints as recommended in the current guidance documents and ICH guidelines. And an informative design with randomized group assignments in animals, appropriate controls, masked assessments, adequate study duration, and the evaluation of the product's persistence are needed. Some of the regulatory considerations for clinical development in early phase include dosing considerations. So the preclinical data is used to inform or support dosing used in early phase trials, and looking at potential toxicity, which will be informed through some of the preclinical data. In addition, what is needed is a scientific rationale for dose escalation or de-escalation, adequate characterization of the safety profile of feasible doses, and considerations around delivery devices or the route of administration. For safety, we often look at dose-limiting toxicities, the duration of follow-up, ensuring that it is tailored to individual products. Are there staggering regimens and stopping criteria in place for both individual subjects or the entire study? And to consider monitoring for immunogenicity, evaluations for viral vector shedding if needed, and is there an acceptable safety for the population in the context of this disease or the condition that's being studied? In addition, we have to evaluate for product persistence and think about long-term effects, even in early phase trials. For efficacy considerations, the approval of drugs and biologics must be based on substantial evidence of effectiveness and evidence of safety. And available data has to demonstrate that product's benefits outweigh its risk at the time of approval and throughout the product's life cycle. Certain aspects of product development that are feasible for common diseases may not be feasible for rare diseases. And examples of regulatory flexibility include single-arm studies to support regulatory decisions. In situations where we have a large magnitude of benefit, we often have some flexibility in terms of the framework for the efficacy studies. And well-designed natural history studies can be supportive for rapidly progressing, serious, and rare conditions. We also have to give consideration to any associated companion diagnostics or devices that are co-developed to become available for use with the particular product. And we use expedited programs and other incentives for serious diseases with unmet medical needs. Some of the expedited development programs are listed here on the slide and include Fast Track, Breakthrough Therapy, RMAT designation, which is a program specific for regenerative medicine and advanced therapies in the Center for Biologics, accelerated approval, and priority review. To highlight RMAT, because it is specific to CBER, what is needed is preliminary clinical evidence that indicates that the drug or biologic has the potential to address unmet medical needs for a disease or condition. Now we will talk about further considerations for investigational and approved gene therapies. Gene therapies hold great promise, and over 80% of rare diseases have a known genetic basis. So far, FDA has approved 22 gene therapy products, most of which are for rare disorders. OTP currently oversees more than 2,600 active investigational products, and collaboration and flexibilities are really critical in the development of cell and gene therapy products. This slide just illustrates the tremendous growth that we have seen in the gene therapy space in recent years. Here you see the number of gene therapies that have been approved, and as you see, it also reflects the tremendous growth in the past 5 years. So this is quite a new and evolving field, but it is also quite exciting as we have several more therapies that are in the pipeline for development that truly address unmet needs. This slide highlights some of the recent cell and gene therapy approvals from the Office of Therapeutic Products, and it really does highlight the diverse array of cell and gene therapy products that are being developed. It's quite exciting. In particular, here we have a product from Orchard Therapeutics that was approved in March of 2024 for metachromatic leukodystrophy, a product from Pfizer that was approved in April, which is an AAV vector used for the treatment of moderate to severe hemophilia B, Adaptimmune's product for treatment of unresectable or metastatic synovial sarcoma. We have Autolus's product for the treatment of adults with relapsed or refractory B-cell precursor ALL, and PTC Therapeutics has a product, a gene therapy for the treatment of adult and pediatric patients with AADC deficiency, which is the first product approved for this condition. One of CBER's 2024 priorities, and a priority for the FDA in general, is to further the development of products for rare diseases and for diseases where there is significant unmet need. Out of thousands of rare, hereditary, and acquired diseases, there are hundreds of disorders that are affecting 1 to thousands per year that could be addressed with novel gene therapies. And addressing molecular defects may reduce some more common diseases to very rare diseases. Actions at the Center for Biologics to address rare disease development include advancing manufacturing technologies for cell and gene therapy through research, applications of the platform technology provision, which will allow us to leverage information across products to expedite product development, to work to really clearly define the use of accelerated approval for gene therapies, exploring concurrent submission and product review with other regulatory authorities, and communication pilots for rare diseases. One particular effort in the Center for Biologics to highlight is our recent COGENT pilot, or the Collaboration on Gene Therapies Global Pilot, to help accelerate the development of gene therapies. Initial participation will be by standing regulatory members of ICH, and partners may participate in internal regulatory meetings and meetings that include the sponsors. Specific regulatory reviews are also shared across agencies and discussed with our partners. All meetings conducted and information shared is under strict confidentiality agreements, and the goal is really to increase the efficiency of the regulatory process, reducing time and cost for agencies and sponsors. The START pilot is an additional program that is used to further accelerate the development of rare disease products. And in particular, it is to accelerate the pace of development for products that are intended to address an unmet medical need as a treatment for a rare disease. There are 4 CBER programs that have been chosen and 3 CDER programs. The requirements for this program are that the product is being developed toward a marketing application. It's intended to address an unmet medical need as a treatment for a rare disease that's likely to lead to significant disability or death within the first decade of life. And with this program, you get enhanced communications with the agency, and additional things such as ad hoc emails or live interactions on an as-needed basis. We are excited to highlight our START pilot selections in the Center for Biologics. We've chosen 4 products, as mentioned. Our first product is a recombinant adeno-associated virus serotype 9 vector encoding a codon-optimized full-length version of human NGLY1 for the treatment of NGLY1 deficiency. Our second product is an adeno-associated virus containing a self-complementary DNA payload of a codon-optimized human ASPAC DNA for Canavan disease. Our third product is a lipid nanoparticle encapsulated mRNA encoding a normal human protein, or mRNA 3705, for isolated methylmalonic acidemia due to complete or partial methylmalonyl coenzyme A mutase deficiency. And our fourth product is a recombinant serotype 9 adeno-associated viral vector, MECP2 transgene, for the treatment of Rett syndrome. The ARDEA pilot program is an additional program that seeks to advance rare disease drug development by providing a mechanism for sponsors to collaborate with FDA throughout the efficacy endpoint development process. Admitted sponsors may request up to 4 meetings with FDA to discuss development of their proposed novel endpoints. And after completion of 4 ARDEA meetings, the sponsor can request additional input from FDA as needed through other formal meeting mechanisms, such as Type B, Type C surrogate endpoints, or Type D meetings. This program is across both the Center for Biologics and the Center for Drugs. So in conclusion, I'd like to thank you for your time and attention. I've included several OTP resources here, which include guidance documents, our OTP events, meetings, and workshops webpage that lists upcoming events, and links to information and recordings of past OTP events. Interactions with the Office of Therapeutic Products website, which has a lot of information, and in addition, our OTP Learn webinar series. Thank you for your time and attention. And listed below is our contact information. Thank you again.
Well, thank you very much to Karen and Nicole for excellent lectures. Karen for covering in detail the physiology of mRNA vaccines, the general place, the context in which they're developed, and the enormous numbers of possibilities that they may address. To Nicole for introducing us to the enormous spectrum of opportunities with gene therapies and other innovative therapies. I know that Nicole is under a little bit of time pressure, so I've got several questions here, and I was going to begin with her questions. Yeah, there's a nice broad question for you here, Nicole. In your opinion, which is the most important challenge that FDA is facing regarding regulation of gene therapies? What are your opinions there?
Yeah, thank you for that question. It's an extremely important question. So, gene therapies, they provide a lot of opportunity to address significant unmet need in rare disease. And so I think one of the challenges is really the balance of getting therapies to patients that need them as in an expedited fashion as possible, while also, maintaining all of our standards that are quite necessary in terms of safety and efficacy. So one of the challenges I would say is, because we have such targeted and directed therapies, that we can get things to patients sooner, but there's significant heterogeneity within, in particular, the rare disease space. So we really need to be able to follow patients after approval for longer term studies, and really getting that commitment and truly understanding how to do long-term evaluation in the post-market setting effectively, so that we can continue to monitor and to continue to see how these therapies work over time, while also maintaining our commitment to getting things to patients as soon as possible. So a lot of challenge, but a lot of opportunity and a lot of exciting products and a lot of exciting work right now in the field.
Thanks very much. I think that's an excellent overview of the challenges. And I would imagine from my drug perspective, another challenge is that we just don't have that many patients to work with. So I think we're certainly sympathetic to the environment in which you're working under, with really limited number of patients and novel technologies. I had another question for you, Nicole, which was, in the Office of Therapeutic Products, is CAR T included? Perhaps you can just expand a little bit on what your approach is to CAR T.
Yeah, so we do have CAR T cell therapies within the Office of Therapeutic Products. That's where they are regulated. And I will say that's also a really exciting field. The CAR T space, as many know, has really expanded in recent years to not only include malignant conditions, but non-malignant conditions as well, and provide a lot of hope in a lot of areas that are really in need of more targeted therapies. So yes, they are in our office, and we do see quite a few INDs in this space that are very across the spectrum.
Great, thanks very much, Nicole. I'm going to turn to Karen now with a couple of questions, and there's one question which is broad and I think makes a good beginning to the discussion. Karen, the question is, why is it hard to make vaccines for some conditions?
Thank you. You're right. It's a great question. Many factors, really, and one of the great examples is HIV. Why after so many years we don't have a vaccine for HIV? And it depends on the biology of the viruses. In this case, HIV, you would really need to prevent infection, not only disease, and that's very hard to do. It also depends on whether we understand the virus science enough, or maybe if a clinical trial is feasible, is if you have a reasonable clinical endpoint that you can measure the effect of a vaccine. So there are many, many reasons why there are some diseases that don't have vaccines, even though they have a great burden, from scientific all the way to practical reasons.
Thanks. I think an excellent answer to a question that faces all of us in the medical community. So a couple more questions for you, a lot of interest here. One question is that vaccine typically requires assessment of biological potency at release and for stability. And the individual is asking, how is this performed for mRNA vaccines?
Another great question. Very good audience today. So that doesn't depend on the platform. Usually potency depends on the disease and the pathogen that you're studying. So your potency assay, whether it is an animal model or immune assay or whatever else that you're going to do, depends on, or a correlate on what pathogen you have that vaccine against. So that's going to determine the potency assay that you're going to use, which is, can be used for mRNA as well.
Great, thanks for that. And keeping you in the hot seat here, there was a technical question which perhaps you can answer. Were pseudouridines or other modifications recognized as reactogenic causes of adverse events during the development of the mRNA vaccines for SARS-CoV-2?
Excellent question, and sorry I did not clarify. No, all that science was done years before. We were very lucky that it was all done years before the pandemic. And of course, mRNA is used for many other targets. One of the most important ones is cancer. So there was a lot of data accumulated and a lot of science behind pseudouridines way before the SARS-CoV-2 pandemic.
Thanks very much. Nicole, if you're still on, there's a question for you. For gene therapy, how much of a barrier is pre-existing immunogenicity?
Yes, so immunogenicity is something, as someone astutely has made that observation, is something that we really do consider and think through a lot. And in really some of the early phase gene therapy trials, we had a lot of lessons learned in terms of immunogenicity. So in the gene therapy space, in general, a lot of sponsors and companies and even academic investigators are addressing these potential immunogenicity issues to the transgene, in particular, with immune medications and other things around the administration of the gene therapies for a given amount of time to minimize the immunogenicity risk. So those risks are, as you point out, those are things that we consider in the development of trials and also in the development in this space. And we do have ways to try and mitigate those risks, and then also to follow patients should this occur. So it is something that a lot of consideration is given to. I wouldn't say it's a barrier at this point, because a lot of people are addressing it in a way that does help with safety.
Thanks very much. A very good answer to a tricky question. I guess we have to be as creative as we can to make a question for Karen, which is close to my heart as an infectious disease doctor. How can mRNA vaccine technology contribute to the fight against antimicrobial resistance? And what potential does it have in preventing infections caused by drug-resistant bacteria? Any thoughts there, Karen?
Yeah, that's a tricky question. So mRNA, I mean, it might have a lot of different approaches. The one I know about, because that's not really my area of expertise, is I know we're trying to figure out how to deliver monoclonal antibodies through mRNA, which is being studied in several areas. And that, I imagine, will be an approach that could be used for AMR, but I'm sure others, maybe even Nicole, has more information about that. But it's a great question.
Thanks for that. Karen, I had another one for you, and I think we're approaching sort of the end of the session. In your presentation, you've mentioned multiple times that mRNA vaccines have been successful. Are there no safety concerns for mRNA vaccines, especially with COVID mRNA vaccines? So this is, again, about safety, which you did touch on in your talk. Any thoughts about that?
Yeah, and thanks for the opportunity to clarify, because yes, COVID vaccines were very successful. As you saw, some of the data, we prevented a lot of deaths and a lot of hospitalizations. But of course, any intervention, anything you take has a risk, and vaccines are not different. And we always evaluate the ratio of benefit risk. So in a way, that's the end point, evaluating the benefit against the risk. And of course, we continue to evaluate the safety of any vaccine, including mRNA vaccines and COVID vaccines, even post-marketing or post-authorization. So we are continuing today and in the future surveying safety events and informing the public of those events. So that's an ongoing activity.
Well, thank you very much. I think that brings us to the end of the session here. It's been a long and very engaging day. Again, very grateful to our speakers from the Center for Biologics on two areas of major interest. And at this point, we'll be closing for the day. I just wanted to let everyone know that tomorrow we are shifting phases, and we'll be talking primarily about investigator responsibilities and about FDA and our regulatory role as to ensure the quality of clinical trials and the data we receive. So we very much hope that you'll be able to join us for tomorrow for these very sort of practical aspects to clinical trials and investigator responsibilities.